Why are we working on epithelial barrier theory? That's an important point. In 1998, we started with Axel Trautmann on epithelial cell death and the reasons for epithelial barrier disruption in skin, lungs, and sinuses, which was demonstrating the first mechanism of eczema and at the same time epithelial shedding from the lung bronchial mucosa or also from sinus mucosa. And at that time, we developed artificial skin equivalents. In 2006, we better understand the major functions of epithelial barrier, keep away functions like a physical barrier, laminar reticulars thickening, and IgA, wash away functions such as mucus, ciliary movement, barrier opening, and drainage, and an expulsion response and suppression functions. And we understood between 2009 and until now that the immune system makes a very strong triangle with the epithelial barriers and microbiome, and T cells, type 2 innate lymphoid cells, and IL-13 play major roles in this. And we can also understand that environmental agents that are seen in this picture, like laundry and dishwasher detergents, surfactants, diesel exhaust, ozone, nanoparticles, enzymes and emulsifiers in processed food, shampoos and body cleaners, household cleaners, toothpaste, particulate matter, and microplastic damage the epithelial barriers. The epithelial cells are not touching each other. There is microbiome in between, and some of the microbiome has passed. This is a translocation of the microbiome, and the leaky barriers allow the passage of allergens, toxins, pollutants, microbiome, and opportunistic pathogens, and these agents show synergistic effect in breaking the epithelial barrier. We have to quote shortly here Paracelsus. Paracelsus was the pharmacist of Canton Schwyz in Switzerland, and he said, "Sola dosis facit venenum." That means the dose makes the poison. Everything can be poisonous if the doses are high enough, and there's no poison without the dose. So that's a very important concept to understand. And the substances that we are talking about, they all have a poisonous dose and they all have a non-toxic dose. So that's an important point. Now, to start with, we will talk about detergents, surfactants, and type 2 response. Here, the main culprit is sodium lauryl sulfate. Sodium lauryl sulfate was discovered during the Second World War, was introduced to detergents in 1960s, and it became one of the main substances in detergents, in shampoos, household cleaners. I was looking at a detergent, it was containing about 5 to 10% sodium lauryl sulfate. And in household cleaners, we see it. In some toothpaste, there's 3% sodium lauryl sulfate. And cosmetics and skin cleansers, there's 0.5 to 2%. It can be extracted from house dust, pillows, and bed sheets. Now, this is an experiment that by Ming Wang that shows the laundry detergents, but similar experiments were done with also sodium lauryl sulfate. And we can see here 1 to 50,000 dilution causes a big regulation of the cellular mechanisms. And here is RNA sequencing, and the upregulated group of genes are lipid metabolism, oxidative stress, cell survival, and interleukin-33. And downregulated group is cell adhesion, extracellular matrix organization, and wound healing. The interpretation of these findings is that there is a chronic wound that tries to survive the cells. And main culprits are sodium lauryl sulfate and sodium dodecylbenzene sulfate. This is a very interesting, very exciting experiment by Hideaki Morita's group and Kenji Matsumoto from Tokyo University. And here, 4 drops of detergents are put in the nose of a mouse, 0.1 and 1%. And we can see here the control mouse, a very healthy, normal bronchial mucosa. You can see the 3 bronchus here. And here, there is a very severe mixed inflammation, eosinophilic and also neutrophilic inflammation. Total leukocytes increase with the increasing dose of detergent. Eosinophils are responding very strongly and with 1% of the detergent, and neutrophils are also increasing very, very strongly. Then it was possible to look at which cells are responding to the sodium, and we can see that innate lymphoid cells, mainly type 2 innate lymphoid cells, are responding. 44% are becoming positive for IL-13 is happening. Basophils and T cells are not responsible for this early response. And this is the resting condition, and in IL-5 making, type 2 innate lymphoid cells are 17%. So here we see the increase with detergent total leukocytes. Eosinophils are increasing, and neutrophils are increasing. But if there is a REC2 and common gamma chain knockout that doesn't have any innate lymphoid cells, then we see there is no increase in eosinophils. So that's very important to show that innate lymphoid cells are responsible. Another important factor is IL-33. And here we can see with detergents, IL-33 staining in the mucosa very significantly. In the control, there's very few cells that are positive. And we have here an IL-33 knockout mouse, and we can see that IL-33 knockout mouse has a very significant decrease in eosinophilia. That was increasing by normally detergent exposure, but when IL-33 is negative, is not expressed in the mouse, then we can see that its eosinophils are showing very significant decrease. Now we have another important study from the Mayo Clinic group, detergents and sodium lauryl sulfate and the relationship with eosinophilic esophagitis. We said that eosinophilic esophagitis started to increase after 2000, and it came from very few cases to several 100 million cases. And we can see that esophageal barrier is broken by sodium lauryl sulfate. This is shown as sodium dodecyl sulfate. It's the same material with 5 microgram per ml doses. And we can see that IL-33 showed a very significant increase in the dose of 5 microgram per ml and 10 microgram per ml. And here we see in the mouse, a normal mouse esophagus, and we can see here sodium lauryl sulfate. This is 0.5%, and here it's a very significant eosinophilic inflammation with the thickening of smooth muscle, mucosal thickening, and also the lumen is full with stricture and fibrosis taking place, and there's a lot of eosinophils, and this eosinophilic infiltration and also lymphocytic infiltration is taking place very significantly. Then from here, we come to very shiny dishes. This is a study done in our group. We can see professional dishwashers. Professional dishwashers were introduced to our lives after 2000, and laundry, normal laundry, and also household dishwashers were introduced earlier in 1960s. Professional dishwashers have a rinse aid, which contains alcohol ethoxylates, and Ismail earlier did these studies showing that up to 20,000 times dilution, the cells, these are the gut also epithelial cells, and they are dead, and in 40,000 dilution unexposed, we can see that the cells are alive, and the rinse aid is responsible for the cell death. Detergent itself is not. And here they were able to isolate these detergents from the cutlery, dishes, and glasses, and show that there is a cytotoxicity in 1 to 2 dilution, 1 to 5 dilution, 1 to 10 dilution, more than unexposed. So they were able to show that this extracted detergents from the cutlery and glasses and also plates was causing very significant decrease in transepithelial resistance. That means the barrier was opening, and this is also shown by passage of the FITC labeled dextran in 1 to 2, 1 to 5 doses. This is the cover of our journal about also eating and habits are changing in our life. I'll talk about now packaged food, mainly emulsifiers, preservatives, and surfactants. And packaged food became a very important way of feeding in our lives, mainly in the last 20 years. And before 20 years, the shelf life of packaged food was around 1 week in the big markets in Switzerland. And now the shelf life of packaged food is up to 6 months to 1 year. The commercial aim was to extend the shelf life and prevent the separation of the liquids. And it was successfully achieved from the commercial point of view, and shelf life was increased by several months. The mechanism and the aim here is to add the preservatives and emulsifiers to kill fungal growth and bacteria and provide a thickening effect. We asked them the question, is it possible to do this without harming human cells? Is it possible to use these preservatives which have antibiotic effects or emulsifiers which keeps the water inside? No, it is not possible. It's practically impossible. So Ismail, again, with our group in the epithelial barrier research, focused on the commonly used emulsifiers, 2 of them, P20, Polysorbate 20, and Polysorbate 80. Polysorbates are twins that if you are doing a gel electrophoresis, you are using twins very commonly. And twins are very soapy, very toxic, and we can see here that the toxicity goes down to 0.1%. But very excitingly and very interestingly, 1% of these polysorbates are allowed in the ice creams and pastry in the United States. This is a huge public health problem, of course. And in Switzerland, you cannot use polysorbate in ice creams, but you can import ice creams from Denmark or from United States and sell them in Switzerland, which contain 1% polysorbate. So here's a big, long list of emulsifiers. And we can see that there's many of them, and you can read the packages and labels of the packages. You can see that there's a long list of emulsifiers, and currently we are working on all of them. So if we are talking about opening of the tight junction barriers, here we can see from the apical side, from the top, that the barrier is sealed. This is a closed tight junction barrier, very significantly. And this is an open tight junction barrier, which we first showed in 2010 by Maciej Chalubinski.